Dr. Hernando Bacosa is a postdoctoral fellow at the University of Texas at Austin Marine Science Institute. He has a strong interdisciplinary background, and his research interests are at the interface of environmental chemistry and microbiology, particularly on understanding the interactions between microbial communities and environmental pollutants. His current work on unraveling the roles of bacteria, phytoplankton, and environmental factors on the fate of oil in the Gulf of Mexico is funded by the Gulf of Mexico Research Initiative through the Research Consortium on the Dispersion Research on Oil, Physics and Plankton Studies. Dr. Bacosa received his BS in biology with a major in ecology from Mindanao State University in the Philippines. He completed his MS and PhD in environmental sciences major, environmental chemistry and microbiology from Tohoku University in Japan. Dr. Bacosa is also an expert gardener and an avid traveler. Let's welcome our guest speaker.
Hello. Thank you so much, sir, for the kind introduction. And it is an honor and privilege to be here this afternoon to talk about what I have been doing in the University of Texas Marine Science Institute in Port Aransas. And today I will talk about my work on the photooxidation and biodegradation of spilled oil in the northern Gulf of Mexico. Let me talk first about UTMSI. So UTMSI is located in Port Aransas, and this is our campus. We have the research facilities here. We have the peer lab here. We have the animal rehabilitation keep, and we have the marina for sampling. So UTMSI is dedicated to understanding and sharing our knowledge of the world's oceans and coasts, which are critical ecosystems for the health and future of the humanity and the earth. And this institute is actually the first marine lab on the Gulf Coast, established in the early 1940s. And we also manage one of the biggest estuarine reserve in the nation, which is the Mission Aransas National Estuarine Reserve. And we have 14 faculty members of different expertise. We have about 30 graduate students at a time. Then we have 80 support staff, which are the backbone of the institute. And then we have a diverse research area. So I'm working on oil spill. Some people are working on climate change, red tide, drug development, larval fish ecology, fish migration, and a lot more. And yeah, as what they mentioned, I'm from the Philippines originally. So I spent my first 25 years there of my life. And I grew up as a strong boy. And then I moved to the land of the rising sun, where I spent my 8 years of my life there. I mean, a different country, different culture, very silent culture. They don't talk that much, they're very silent. And then I came to this very, loud culture. So it's like really a different culture for me. But the Philippines is somehow in between. So we have basically like an Asian culture. Then we were colonized by Spain for 300 years, by the US for 40 years. So I mean, it's nothing new. Just I got 1 star, but we have 3 star in the Philippines. I didn't get the sun though. Okay, so in the Philippines, when I was in college, I studied about mangroves. Who among you here know about mangroves? Mangroves? These are just trees. I mean, if you travel along the coast, you can see these small trees. They're actually shrubs, and they're actually very critical for the protection of the coast. And they are also serve as fish laying for the fish. I mean, they protect the fish, and this also provide protection for the younger fishes. So in the Philippines, we have lots of oil spill also, and among them are the mangroves. So when the mangroves are affected by oil spill, these small animals, organisms, we call it meiofauna. They are actually about 1 millimeter in length, but they are very important in the recycling of nutrients and decomposition of the leaves. So when there is an oil spill, their population actually is reduced by almost 70 to 80%. And therefore, the recycling of nutrients is being affected. And I'm really interested about mangroves. So when I went to Japan, I forgot about my research because I enjoy Japan. It's a very beautiful country. You know, I enjoy climbing Mount Fuji. You know, like walking in the Japanese garden, snowboarding and skiing. I love it. And eating Japanese food. Everything is raw, sushi, sashimi, seafood. And yeah, it's a beautiful country, very high tech, and high tech in the city, but it's very conservative on the countryside. It's a country of contrast. But of course, I didn't forget my study. So I worked also on mangroves there, but this time working on the bacteria in the mangroves. So in the mangrove sediments, you have those bacteria that are capable of degrading the oil, and in southern Japan, that is actually a very semi-tropical environment. I isolated these different bacteria. So I mean, they are different bacteria, and I made like an effective oil-degrading bacterial mixture that could be used to clean up an oil-contaminated site. And here in UTMSI, when I moved here in 2013, April, I started working on the fate of oil after the Deepwater Horizon oil spill. Who among you here know about this Deepwater Horizon oil spill? Only very few? Okay, so I studied photooxidation and biodegradation. So photooxidation, it means sunlight, okay? The sunlight affects the degradation of the oil. And when you see bio, it means life. It's the bacteria that are actually eating and degrading the oil, like the way we obtain energy from the bread and the food we eat. Okay, first, I have a question. I need audience participation here. Okay, question number one. What company claims responsibility for the Gulf oil spill or the Deepwater Horizon oil spill in April 2010? Wow, very smart people. Okay, next, BP, okay? How long did it take to finally cap the oil that was spilling in the Gulf? A, raise your right hand. B, oh my God, okay, so it's 3 months. Okay, so the spill was on April 20th, 2010, and it was finally capped in July, around mid-July. So it's about 90 days of spilling, like releasing this oil. And 11 people died in the Deepwater Horizon explosion. Yes? True. Okay, you have the basic information. I just want to make sure that you know this basic information. Okay, it's true. So the Deepwater Horizon oil spill is, it happened on April 20, 2010, spilling about 210 million gallons of crude oil in the Gulf of Mexico. So the affected area is like about the size of the state of Virginia, 43,000 square miles. And some of those oil, like 5 to 10%, were found in the seafloor. That's about the land area of Corpus Christi. And the sediment seafloor have some benthic organism in there that were actually affected by the oil spill. And then the coast, the shoreline, were coated with oil and tar balls. That's about 1,300 miles from Texas to Florida. So it is the biggest oil spill, the world's largest accidental oil spill, and an unprecedented disaster in the history of environmental science in the United States. And after that one, I think a few months after the spill, BP, they commit like $500 million to fund research about the effect of oil spill on the environment. And with that $500 million, a certain board, a board was established, and they named the board as the Gulf of Mexico Research Initiative. So this board actually administered this $500 million for 10 years, and it distributes this money to universities and research institutions along the Gulf to study how the oil is being degraded and how can we avoid the spill, and how can we develop some technologies to have a better preparedness in the future. So basically, the board study the effects. They provide funding for these institutions to study the effects of Deepwater Horizon incident and the potential associated impact of this and similar incidents on the environment and public health. And also it develops some improvements for spill mitigation, oil detection, and characterization, and advanced remediation technologies. And to do that, it funded like 12 research consortia. When you say research consortia, it composed of different institutions, multi-institution, multidisciplinary. So I belong to this consortium, the DROPS. It's the Dispersion Research on Oil, Physics, and Plankton Studies. So the leading institution is the University of Texas Marine Science Institute. Actually, we got like $10 million for 3 years. And yeah, so we have ecologists, we have chemists, and simulator and modeler from UTMI. We have the plankton ecologists from University of Wisconsin. We have the mechanical engineers from New Jersey Institute of Technology. We have the chemical engineers from Texas Tech. We have the mechanical and biochemical engineers from University of Pennsylvania. We also have the mechanical engineers and some chemical engineers from Johns Hopkins University. And we have medical doctors and medical researchers from Johns Hopkins University. And SINTEF is actually the biggest research institution in Norway. And we have people who are doing some numerical simulation and modeling. So this is really a multi-institution and interdisciplinary research group. So, and it's really a nice environment to learn something new, something different each day. And I'm excited to be a part of this group. So this is our team. That was actually 2 years ago. I'm still small, but you know. I think I have some bacteria in me, okay. So, but now, yeah, our group is much bigger. So I think we are more than 20 this time, including some students and some researchers. And yeah, we do a lot of research from dispersion and dilution of petroleum under the action of physical and chemical processes. We have the chemical evolution of the oil, like how the oil changes from the oil spill site when it reached the beach or when it formed tarball. And then we also study how the bacteria and how the plankton affect, you know, the disappearance of the oil. And we also study how the oil, especially the aerosols, affects human health. So this is the primary concern of people in Johns Hopkins the medical team. And then we also focus on some mesoscale and mesocosm. So we build like tank to simulate, because to simulate how the oil was released from the seafloor, and then as it ascends to the surface, how does it change, you know, depending on how it become in contact with, say, phytoplankton and zooplankton and bacteria. And one thing with this spill is there was an application of dispersant. So this dispersant Corexit, so it's basically how when you apply like a washing detergent to your oiled dishes, so it produced like a small droplets of the oil. And when the oil forms small droplets, it can be easily degraded by the bacteria. And this Corexit actually is of unknown composition, because the company did not release what is the composition of this. This is new to the environment, and we don't know whether the bacteria can degrade them. So, and they say it could be, you know, plus, minus, or zero benefit from applying this one, because it can degrade the oil, but it could be more toxic to the small larvae of the fish, more toxic to the phytoplankton, or more toxic to the zooplanktons. Okay. So this oil spill happened like 1,300 meters down there, like on the seafloor. So, and after that one, there was a formation of this 35 kilometer of plume of oil and dispersant. And without this dispersant, actually, the oil is supposed to go up, because you know when the oil comes in contact with the dispersant, it produces an emulsion. So, and that emulsion stays here. So that is the plume. But when they apply dispersant here, you can see that some oil remains in here. And this is a very cold environment, 5 degrees Celsius. So the bacteria, you know, when you put your food in the fridge, the bacteria are not active, right? It can stay for 1 week. But when you put your food on the table, after 1 day, you can You can smell the good smell. So basically the bacteria are active at higher temperature. But at lower temperature, like your fridge, they are not active. So the scientists were actually questioning this plume. They say that the dispersant is not really effective because it have provided more damage by the formation of this plume. And the damage is really unprecedented in history. Yes, sir?
So I'm just curious if you know of Terry Hazen's work.
Yes.
And he found a new psychophilic bacteria at those depths.
Yeah.
He found that they were actually degrading.
Yeah, they are degrading. So you have unique bacteria in there. And in the surface, you have different bacteria. So before they didn't know that there are bacteria there. But when they did some sampling, they found out that there are cold-loving bacteria that are really degrading the oil. Yeah, that's amazing on how nature works and how nature heals itself. Yeah, okay, unprecedented environmental damage, destroyed marine life, turtles, birds, invertebrates. It is destabilized marine communities and populations. It's degraded shore amenities. Of course, no jobs for fishermen. Impact on human welfare. You cannot go swimming, you cannot go fishing, especially when you were in Louisiana or Alabama or Mississippi. At Texas, I think we were luckier that time because we were really far. I think about 400 miles away. And then with these 210 million gallons, they say they collected about 25%. 17% were collected, 5% were burned, 3% were skimmed from the surface. But 75% is unaccounted for. The question is, where did the missing oil go? 75%, that's about 150 million gallons. Can you imagine that one if you can put in your gasoline tank in your car? It can last forever. Okay? So, before answering this question, we have to understand what is a crude oil. So, a crude oil basically is, let's see, it's a droplet. And then inside the droplet, you have the aliphatic hydrocarbons and the aromatic hydrocarbons. I think if you have taken an organic chemistry class, it's just basically a carbon and a hydrogen. So for the alkanes, you have this chain. It could be 2 carbon, 3 carbon. It could be branched like this. And then you have the aromatic hydrocarbons, which is the benzene ring. Usually the benzene ring is the smell that you smell from from the gasoline. It's aromatic. So the base is like an aromatic ring, and then when they fuse together, you can have 2 rings, 3 rings, 4 rings, 5 rings. You call them polycyclic aromatic hydrocarbons. And these are the components that are of primary environmental concern because they could be carcinogenic or mutagenic. Especially these betics, when they contaminate the groundwater, you cannot actually, you cannot get anything there. And in Texas, you have lots of environmental regulations. And there are certain concentrations for this. And for the alkanes, if you have 6 carbon, that's C6. If you have 10, that's decane, C10. And if you have 16, you just combine hexadecane. So that's the way. And then if you have longer chain, like if you are working with your engine oil, you have like C40, C50. So it's just a chain of this carbon and hydrogen. And then for the PEH, the U.S. Environmental Protection Agency has 16 priority PEH because they believe to be a potential carcinogens and mutagens. So there are 16 of them. Naphthalene is a component of a mothball. Then you have 3 rings, 4 rings, and you have up to like 6 rings. So this is usually the primary indicator of like environmental pollution, especially of like due to oil spill. So if you analyze by gas chromatography, you will see these peaks. And gas chromatography is one of the techniques. It's the most common techniques to analyze oil. You analyze the hydrocarbon, all the alkanes and all the PEH. But of course, it has some limitations. And based on this analysis, there could be hundreds to even thousands of these hydrocarbons that you cannot actually determine. It's impossible to determine all of these compounds. And if you can look at this, if you have your gasoline, So this means that as you go here, as this is a time, so lower molecular weight hydrocarbon elutes first in the column. So these are lighter, and then you go to heavier. So gasoline, you have more. I mean, the higher is the peak, the more compounds you have. So you have this butane is C4, pentane is C5. So you have a lot of lower molecular weights. Like for gasoline, it's about C10 below. If you go to kerosene, you have more here, C8, 9, 10, 15. If you go to light oil, so you have C26, C25. And if you use your engine oil, you change oil, those could range to C30, C40, or C50. So it depends on the property of the oil. And of course, the behavior of the oil also depends on the kind of the oil that you have. If you have a gasoline in there, it will evaporate in in a day. Like 60% will be gone, especially in a very hot summer. But with like the heavy oil, it could last for decades, and it can form some tar balls. Okay, so there are several factors affecting the fate of the oil in the environment. It could be like photooxidation. Oh, it works now. Photooxidation, and then some of the, like the C8 below, C8 or lighter, will evaporate. Some of them will be emulsified because we have lots of compounds in the environment. Some of them will be dispersed. Some of them will be absorbed to some particles. Like we have lots of floating particles in the ocean. Some of them will be absorbed to some, plankton. And some of them will be biodegraded by bacteria in the sediment, bacteria in the water column, bacteria in the surface water. But, it's not easy to determine this one. That's why studies are now quantifying, because the Gulf is a unique environment. If that spill happens in Alaska, it could, because it's a very cold environment, that could last for like decades or centuries. But the Gulf is a very dynamic system, high temperature, intense sunlight. That's why, after 1 year, the oil is gone. But some of them actually are buried in the sand, in the sediment. And this is a very complex process. So we are trying to understand what are those processes that contributed to the disappearance of 75% oil. Okay, the key elements affecting rates of degradation are the oil, the composition. Okay, if you have lighter oil, smaller molecules, they escape, they evaporate, they can be easily eaten by bacteria. And for the biotic factors, if you have a very hungry bacteria in there, they could easily eat that one. Grab it, and they can transform into, they multiply, and they become very abundant. Because the Gulf of Mexico is really a hub of oil-degrading bacteria. You have the natural sieves in there that has been there for decades or millennium. And they say that the oil in the Gulf are really used to eating the oil. I mean, these bacteria are used to eating the oil. That's why after the spill, the bacteria can easily eat it because they get used to having oil as their food. And we have the abiotic factors, temperature. I mean, the warmer it is, the more evaporation, the more active are the bacteria. Nutrients, if you have more nutrients, because degradation of oil needs some nutrients. And then sunlight, and salinity and others. So bacteria is very simple. We have bacteria all around, wind, in your body, soil, wherever. So they are single-celled organisms, and they are the most successful or living things on Earth. They conquer all ecosystem, human body, oceans, even the sea floor. They are everywhere. And they are capable of degrading the oil. So if you have the oil droplet here, this is an example of Alcanivorax, an alkane degrading bacteria. So this bacteria have the enzyme to degrade the oil. Like we have the enzyme to digest carbohydrates. What enzyme is that? Thiolin? Amylase? Something, it's a lipase, so they have that one. So once the oil will trigger some response for that genes to be expressed. They surround the oil, and then after that one, it goes to the cell membrane, and then there should be an attack by oxygenases. So they need oxygen. And then, after it goes to different pathway, and finally, it will be transformed into carbon dioxide and water. And some of those will go to the biomass, and some of those will go to, they will reproduce. So oil will be the source of energy for the bacteria. The way we eat bread, meat, or whatever. And degradation of bacteria doesn't need one. Degradation of oil doesn't need one bacteria. It needs like a community, different kinds of bacteria. Because in nature, we have thousands or tens of thousands different species of bacteria. So it is actually like, for example, the first bacteria will degrade the oil, and then the oil could be transformed into an alcohol. It could be transformed into an aldehyde, into an acid. Then another bacteria will consume that alcohol, transform it to acid, and then finally another bacteria will take care of the final conversion to carbon dioxide and water. It's a very complex process. If you combine two bacteria, it's simpler. But if you are working with water sample, sediment sample, it's really impossible. But now we have really good technology to analyze it. We have metagenomics, and we have some metatranscriptomics. Those techniques are really, really helping us to understand this process. Okay, so biodegradation is, a product of like a community of microorganisms. And the warmer the water, the higher is the degradation of the oil. And in our study, we try to understand like the biodegradation and photooxidation of oil in surface water and the biodegradation of oil in seafloor sediments. So first, allow me to discuss about that of surface water. So what's in sunlight is UV. So that's high energy, and then the same thing, if you have some cell in there, it will create some radicals. The same with oil. So if you have the hydrocarbon in there, that one will be degraded by the sunlight. Intense solar radiation, high surface temperature. And the questions, because in research, we always ask questions. If we stop asking questions, it means we are already unemployed. Yeah, science is always asking questions. If you're running out of questions, you're not for science. It's out of curiosity. So the question is, how does sunlight affect the disappearance of oil? And how does sunlight affect the transformation of oil by bacteria? And how does sunlight affect the bacterial community in the presence of oil, Corexit dispersant, or both? Okay, so to do that, I went to Louisiana, in those bios, and then I took that small research vessel with 15 scientists. What's that? And then we went to the Deepwater Horizon site. This is like a CTD Niskin bottles. This is like used to sample the water, and it can also determine the temperature, the salinity, the oxygen. We drop it there, we got our sample. And then I do some lab experiment. So for my lab experiment, I have the sunlight treatments and I have the dark treatments. So I have 50 ml of water added in these bottles, and then 200 ppm of crude oil, and then a dispersant ratio of 1:20. So we have a control, seawater only. We have the filtered seawater, to get rid of the bacteria. I have the seawater plus dispersant, seawater plus oil, and seawater with oil and dispersant. So there are different treatments. And for the sunlight, we use these quartz bottles because these quartz bottles allows the sunlight to pass through, including the UV. But these dark bottles, of course, they block the UV and the sunlight. Okay, so we incubated them under the natural sunlight in Port Aransas from May to July. You can see the bottles here. And then there is a flowing seawater in here to keep the temperature the same with, with the ambient temperature. And we gather data from 5, 10, 20 to 36 days. And then after that one, we have to extract the DNA from the bacteria. So we are using some kits to extract the DNA. Then after that one, we We amplify them by polymerase chain reaction to make different copies. We target the 16S gene. This is the one that is used to identify bacteria. And then finally, we sequence the DNA. The DNA sequence is just basically ECTG. And now, we are using this spiral sequencing. This is actually, we call it a next generation sequencing that is capable of analyzing a community of bacteria. And for the oil analysis, we extracted the oil from the cultures, and then we used this silica gel fractionation to separate the alkanes and the aromatics, and then analyzed by gas chromatography mass spectrometry, GCMS. Okay, this is the result for the bacterial growth. So we can see from seawater, within 36 days, there is no growth, but you can see the growth of this bacteria in the treatments with oil, with dispersant, and oil and dispersant, suggesting that there is really a very, the bacteria can really consume the oil and the dispersant within 20 days. That's very short. That's really very fast, based on previous literature. Within 20 days, it means that when the oil is there, the bacteria can easily consume them. And no wonder then that these bacteria are the main actors of oil degradation. And if you can see, in the light, you have also lighter oil, and you have darker oil in the dark. And if we look at the oil chemistry, this is the time of incubation. This is the remaining concentration. So if you can see here, for the alkanes, whether in the dark or in the light, they degraded in a similar fashion, suggesting that the bacteria are the one responsible for degradation. If you look at the PAH, this is the light. The light control has no bacteria. The light oil has bacteria. And this one has bacteria too. But with or without bacteria, the PAHs are degraded in a very similar fashion. It means that the bacteria are not playing the role, but it's the sunlight that is actually degrading the PAH. So the bacteria is degrading the alkanes, but it's the sunlight, which is actually a very critical factor for the degradation of the oil. Because, we have an intense sunlight in the Gulf of Mexico. And I've heard that there's also very high rates of skin cancer in Texas. So, yeah, because of the sunlight and the UV. And if we look at the pH, so you have higher degradation for the smaller ones, and then you have like a lower degradation for the heavier ones. And then let's look at the bacteria. So you can see here different bacteria that's in the dispersant with the dark and the light. You can see the difference. So dark and the light, you have Thalassobius in there, you have Alteromonas here, you have Coxilia, and you have Thermos. So you have a different bacterial community developed under the light and those in the dark. And this is a statistical tool to determine, like the similarity of the treatment. So we can see that the dark, they clustered together, but in the light, they are quite apart. It means that light is really a driver of the change in microbial community. And if you look at the oil and oil and dispersant, the same thing. So we have Thalassobius in here, Alcaniborax. You have more of yellow here. You have more of blue here. And then, again, here you have Alcaniborax here and Thalassobius, but you have Alteromonas here and Marinobacter. And if you look at the NMDS plot, the dark is there, the light is there. So light is really a key factor in the development of these microbial communities. Okay. This is another statistical tool. So it's really highly significant, dark versus light. And we have some bacteria that are favored by the sunlight here, and then these bacteria are favored by the dark condition. And you may ask, like, oh yeah, you did your experiment in the lab. How about the thing that really happened in the field during the spill? Well, we can say that our incubation result is very consistent to the field moose and the oil samples floating in the Gulf. So even though our experiment was done 3 years after the spill, the same bacteria were found. The bacteria that were present were very similar to those found during the 2010 oil spill. So it means that the community in the Gulf is really very, very robust and very stable through times. Also, the oil affected the diversity. You can see this one in the light, you have lower diversity. And then in the dark, you have higher diversity. So the light does not only affect the community of the bacteria, but also it affects the diversity of the bacteria. So you have less genus of bacteria in the light, but you have more in the dark, suggesting that the light could have killed some of the bacteria. Because when you expose the hydrocarbon to the light, there is an accumulation of oxygenated compounds, and they could be more toxic. And then there should be a formation of inhibitory metabolites, and one thing is the radicals. The radicals could actually inhibit cell growth, and it could kill them. And finally, it could increase toxicity. And this paper is actually under review. And if you compare all of those things, light explains 50% of the variation, oil is 31%, and dispersant is 21%. So suggesting is light has a more predominating effect on the development of bacterial community. Okay, so for this part, photooxidation is the key process for the disappearance of PEH and very little effect in alkanes. Biodegradation is the key process for alkanes, and solar radiation did not affect the degradation of N-alkanes and did not increase the degradation of PEH. And sunlight reduced bacterial diversity with oil and or Corexit dispersant. And the second part of my talk is about that of the deep sea sediments. So in the deep sea sediments, I'm asking how fast is the crude oil being degraded by bacteria, and how the bacterial community composition and structure change when exposed to crude oil with or without Corexit dispersant. So in the sediment, as you can see, if this is a sediment, the bacteria could be there, and it could interact with oil and dispersant. So I went to another cruise again. It's actually a 10-hour drive from here to Louisiana. Then I just collected this sample. So I took this one from 1,500 depth. It's really deep. So we have to drop this equipment in here and get like this, we call it sediment core. And I just used the top 3 centimeters for my experiment. So we incubated that using crude oil, Corexit, and oil. And I have the control. So the control has no oil in it, and then the treatment of sediment, oil, and Corexit. And then since the temperature down the seafloor is 4 degrees, so I also did my experiment at 4 degrees. And then I have some sampling at a different time after 0.5 month, 1 month, 2 months, 4 months, and 6 months. Oil analysis is the same, fractionation, and then bacterial community analysis. So look at the result. So you can see this one, alkanes, after 1 month, after 2 months, 4 months, 6 months. So you can see in 6 months, about 80% of the alkanes were gone. So these bacteria are really spoiled. They're really spoiled. They're really, really very effective in degrading the oil. They love the oil. And for the PEH, you can see that after 1 month, 70% were degraded. But after that, there was no more degradation. And if you look at the alkanes, you can see that the lighter ones were degraded faster than the heavier ones. Because like when you have a long chain of alkanes, that's too big. It cannot enter the cell. So it needs to be acted by some exoenzymes. But for the smaller chains, like C11 to 15, it can be easily taken by the bacteria. Like when I give you like one big pineapple, can you grab it by yourself? Eat it? Of course, you need to slice it into chunks, right? That's the way, because you need chunks. So these alkanes, these smaller alkanes are like chunks of pineapple. You cannot easily take the whole pineapple. And for the pH, you can see that the same thing. This naphthalene and phenanthrene, the 3 rings were degraded faster, but the bigger ones, like the 4 rings, were degraded like lesser. And then for the bacterial community analysis, you can see from here that this gamma proteobacteria is really abundant in the presence of oil. So you have from time 0, you have this, and then they increase by twice. So if we examine it closely, you can see that from time 0, it increased by more than 20 folds within 2 weeks. And this one is Alteromonodalis. And then the same with Oceanospiralalis. And these bacteria are known to degrade alkanes. Based on Hazen, Dr. Hazen also found that this Oceanospiralalis is the bacteria that love the cold environment to degrade the oil. And then we have another Methylococcales, which also degrade PAH. And then if we look at the Alteromonodalis, we can see that the Colwellia increased by more than 50 folds. And this Colwellia is known to degrade short alkanes and aromatics. And this Alteromonas is the key PAH degrader. And these bacteria also were the bacteria found in the plume during the 2010 oil spill. So suggesting that the bacteria in the sediment and then bacteria in the plume are very similar community, because I think it's like just 100 meters away. Like the plume is 1,300 meters, and then the sediment is like 1,400 meters. So I think there should be some mixing in the seafloor that allow this bacteria to consume the oil. And then to conclude this study, deep sea sediment bacteria degraded more than 50% of pH within 2 months and 70% of alkanes in 6 months. And they have high hydrocarbon degrading ability. An addition of crude oil leads to a dramatic shift in deep sea bacterial communities. And crude oil enriched Alteromonodalis, Oceanospiralalis, and Methylococcales, which resemble that of the plume. So to review our factors affecting rates of oil degradation, we have the oil. We have the biotic factors. The bacteria are responsible for degrading the alkanes. And the sunlight, abiotic factors, responsible for degrading the polycyclic aromatic hydrocarbons. And the take-home message, sunlight has a crucial role in the degradation of aromatic hydrocarbons in surface water. And bacteria are key players in the degradation of aliphatic hydrocarbons in surface water and both aromatic and aliphatic hydrocarbons in deep sea sediments. So sunlight and bacteria are very crucial components of the Gulf ecosystem. And my question number 4, Texas state law requires the companies to report spills within 24 hours, after which state official, how many hours? Who says yes? No. So how many hours? Any guess?
48. 2 hours.
Within 1 hour. So usually like a spill of like I think 1,000 gallons. You have to report that one to TGLO and Coast Guard within 1 hour. After that one, you will pay a penalty. That's really big money. And then question number 5. In 2013, Texas General Land Office or TGLO responded to 40 oil spills in Texas waterways. Who say yes? Yes? No? Yes. Sure? Yes? Yes. No? Yes. Okay, I will answer. No. 423. So you have one spill, like basically one big spill, not like a medium-sized spill, every day. Yeah. That's just in Texas. Can you imagine Louisiana, Mississippi? Actually, you have more oil rigs in there. And yeah, that's the end of my presentation. But before I end, I would like to thank my amazing supervisors in MSI for 2 years, Dr. Zhang Fei Liu. He is a geochemist. And Dr. Dina Erdner, she is phytoplankton ecologist. I didn't work 50-50 for them. I work 100-100. So I was working 200 times, but I got, you know, paid 50-50. And yeah, I mean, I thank different people from this amazing group, from the DROPS Consortium, from the Gulf of Mexico Research Initiative, from the Marine Science Institute, collaborators from University of South Florida, from Michigan State University, and from another consortium. And finally, I thank my family in Port Aransas, the Kilgore family, for like making my life more exciting in the island. And thank you all. Thank you for the blessings. Thank you for the vegetables in my garden. This is my produce in spring. So I have thousands of tomatoes. And they are organic, you know. I don't like chemicals. I don't like oil. But I just study them. So yeah, I have lots of cabbage and broccoli. And if you have questions about gardening, yeah, you're welcome.
Before we ask questions, why don't we thank our speakers?
Yes, sir.
Did you thank BP?
Yeah, thank you for the money. I got my salary there. No, because they say this is like, what do you call it? It's their commitment. Because the damage is because of them. So they give $500 million for an independent research. So we are totally independent from them. And sometimes the findings actually is, you know, BP doesn't agree with the findings of these scientists. So there is like a debate and a conflict on this. Yes, sir.
Hey, great talk. Really enjoyed it.
Thank you so much.
I don't know if you're part of the consortium did this work, but the plume that was at and was staying down there in the Mulder because of the Corexit.
Yeah.
That was, I think, at one point 10 miles downstream from the wellhead. I know that there's multiple fates, but what was the major fate of that? I mean, because I never figured that out. It was submerged, and you showed in one diagram that it came to the surface eventually?
No, it's just stay there. Like it's, so the plume is, look, the 35-kilometer plume. And I think there are a smaller plume, but the main plume is 35 kilometer of oil and dispersant. So, I mean, it's the first time in history that they found this one. And they were thinking that it could stay there forever because, you know, that's a very cold environment and bacteria are not active. But scientists began sampling from the plume, and they found that these Coluelia are cold-loving bacteria that loves to eat the oil. And yeah, and after 6 months, the plume is gone. Because these bacteria also are get used. Because I told you that there's natural seep everywhere in the Gulf. And so the bacteria are get used to, you know, eating the oil for, I don't know, thousands of years or hundreds of years. And they really, you know, they really love the oil.
They were feasting.
Yeah. Yes, sir.
Can this information be found on the website?
Yeah, I publish my work. So our job is to write a paper and submit for publication. So yeah, I mean, if you search my name, you can find my list of papers. You can download my paper. But of course, if you don't have an access, you can just email me. But usually, the Gulf of Mexico Research Initiative posts this one in the website in a very simplified form. Because we write like 10 pages, 20 pages research article. That's very difficult for a common person to understand. But you know, they make like one-page article for the public to know about this one. Everything will be available in the website. Yes, sir.
When you use the term degradation of aliphatic hydrocarbons as well as polynuclear aromatic hydrocarbons,
Yes.
what is the threshold for degradation?
Oh yeah, that's a very good question. So as I've told you, it's really, oil is a very complex, mixture. Hundreds to 100 to 1,000 compound. So we only have this GC amenable compounds. So usually it's just 10 ppm in Texas law, I think. So, but when we can see like 90% is degraded, that's amazing.
It is.
But I think for the environmental, environmental standards, it's really, more strict. Like it's 10 ppm, I think, for the oil. That's a total oil. Because we cannot quantify everything, it's the total hydrocarbon. We call it total total petroleum hydrocarbon. 10 milligram per liter.
Thank you.
Yes. Questions from students? You should ask questions.
It seems like in both the control and the ones that you added the oil and the exo to, the bacteria levels spiked in both. Could you explain that?
Oh yeah, there's an increase of bacteria when they added the oil.
Yeah, but in the controls it seemed like there was also...
Oh yeah, wow, excellent question. Yeah, I should have explained that one. Because I'm always aware of the time. I 8 years in Japan. You have to be in time. You have to come 30 minutes before presentation, and okay. So what he's talking about is in the control. You can see that one in the control. There was also an increase. See? It's because the control have some oil. You have some hydrocarbon in there. So my sediment sample has some like 120 nanogram per gram of pH and alkanes. That's why those bacteria also increase, because you have those oil in the control. But when you added the oil, you have a higher increase. Yeah. It's really not easy to simulate the environment. Yeah, but it's just the control will provide you some explanation. Yeah, question from, I think we still have 5 minutes. Students. You can ask about the garden. Or the worm. I have some horned worms in my tomatoes. Or about my travel. When I was in Japan, I went to, because in Japan, they sent us to different countries. It's part of research. We do research. We go to conferences. Yes?
I want to ask about your garden.
Okay.
I just moved into a house, and my parents want to build a little grow box, I guess, a few grow boxes. And when they're trying to, I guess, plow their garden, it's just clay. So I don't know what kind of soil you use.
Yeah, you have to add some mulch, some organic matter. You have the dried leaves and yeah.
Are there like special kinds that you make since?
Yeah, I'm also doing some experiment with my garden. Port Aransas is really sand and very low in organic matter. More questions?
So
Yes, sir.
Out of your research, what you're saying is if there's another oil spill, your recovery process is to introduce more bacteria.
I didn't get your question.
Didn't they submit like $500 million to come up with a way to recover and prevent?
Yeah, so BP, I think aside from the penalty, I think they were paying like 14 billion of penalty, BP, and then this 500 million is just for research.
Right, and that's what I'm saying. As far as your research is concerned, you're saying if another oil spill were to occur, your theory is to introduce more bacteria?
Not really, because that's the technology. We call it bioremediation, adding bacteria. So usually we add bacteria when there's no bacteria in there to degrade the oil. But for the Gulf, we don't advise doing that one, because the Gulf has the oil degrading bacteria and nature can heal itself. So we just need to understand, how it works.
Just understanding what happens.
Yeah, and then
For like recovery.
yeah, and then some engineers are doing the technology. So they develop some adsorbent. When there's oil, they are using some grass or some peat moss to absorb the oil.
So you just wanted to research and see what it did to the environment.
Yeah, for us, that's our goal. For me, as a microbiologist and chemist, as a basic scientist, my role is to unravel what's in nature. But for the engineers, that's their job to develop something.
Okay.
Yeah, and for the health sciences, to examine the health effects of the oil and something like to avoid
So you
getting
guys are
health.
just researching where the 75% went?
Yeah.
Okay.
Currently in my course right now, geology, I understand that there's, accidental spillage and natural seepage from
Yeah.
Earth.
Yeah.
Does the course of action to take also cover the natural seepage as well, or is it just limited to accidental spillage?
Yeah, so now our job is to, mainly for the Deepwater Horizon oil spill, but we study like natural seepage as well. Because, have you heard these tar balls along Padre Island in May or June? So it's really covered with tar balls. And they couldn't really pinpoint the sources of this. And they say perhaps it could be from natural seepage. So we also study that one, natural seepage, and also we sample from, like, have you heard about the Galveston oil spill last year? I was part of that, so I did some sampling there and collected some tar balls. So whatever oil spill, yeah, that's actually part of our research. Yes?
This bacteria that you guys found, I'm just kind of wondering, other than it consuming oil, have you guys found out other things that it consumes in the earth, in the water?
I didn't get your question, I'm sorry. So we found bacteria that consume the oil and then...
Right, have you found out if bacteria consumes something else?
Oh yeah, that's a good question because you have lots of organic matter. You have proteins, you have peptides, you have plant material, you have humic acids in there. So yeah, perhaps we couldn't analyze that one. We only analyzed the oil. Yeah, but they are part of this complex process. So perhaps they're also degrading, compounds other than oil. Yeah, especially when you have when you will sample in Mississippi River, that's really, like you have some dirt in there. So bacteria is really active, not only the oil. But the more organic matter you have, the more nutrients, and the bacteria will be happy to degrade the oil. Because it needs also nutrients to degrade the oil.
Okay, folks, also like to thank Dr. Gardner for inviting us to the post.